Okay guys, so the purpose of this video is to demonstrate the correct way to perform a dihybrid cross in genetics. So let's use simple letters for our example, just A's and B's. So here's one individual, again, is hybrid for two traits. Here's hybrid for A, whatever that trait is, doesn't really matter, and hybrid for B. So in other words, they have both alleles, the dominant and recessive, for two different traits, A and B. We're going to cross the same thing for that parent to make it about as complicated as we can for a dihybrid cross. Here we go. So the most important thing to recognize for this is how to make the gametes correctly, and for that you need the FOIL method from algebra. Just when you thought you'd never need this again. So our first, outer, inner, and last. Alright, but again we're talking about genetics, meiosis, and instead of taking half from each parent this way, which is incorrect, we can't take two A's from one parent and two B's from the other parent. Half means one A and one B from one parent, one A, one B from the other parent. That's how our offspring is going to get the correct amount of DNA. So our first refer to the first two letters that are different. Again, an A and a B to make this correctly. Alright? Big A, big B are our firsts. And again, literally making gametes. Okay? Our outers are right here, the very outermost letters. There's our second gamete. Our inners are right here. And here's the tricky part. We always write capital before lowercase, but that was when we only had two different letters. Now that we have four letters, okay, we want to do it in A-B order, or letter order. If the parents are A-B order, all the gametes are A-B order. Capital before lowercase only comes for this type of an example when you only have one letter. All right, and then our last are right here. Last of each. Again, half of the A's from one parent, half of the B's from the other parent. And since our parents both have the same genotype to start with, we can just copy everything we just did here down the side for our Punnett Square. So there's our first, outer, inner, and last again. Again, parents have the same genotype, and guys, literally, if those are the sperm, these are the eggs. Those are our gametes. So inside of our Punnett Square, we're literally showing what would happen if those two gametes fused. What would the outcome be for the offspring? We're predicting. We're not saying what is going to happen, but what is likely to happen. It's all probability. Alright. So for our Punnett Square, we need to have 4 columns, 4 rows, because we have 4 different gametes for each parent. So that's how we're going to set up our boxes. And again, another trick to remember here, if you have 2 A's, 2 B's for the parents, all of your offspring that are the result of these parents should be the same. 2 A's, 2 B's. They might be different capital lowercase. They will be, most likely. But you want to write them the same order the parents were written. 2 A's, 2 B's. And here's why. There's homozygous dominant for A's, heterozygous for B. The same exact genotype is going to show up right here. Okay? Same genotype here. And this box is when we line up those sperm and egg. However, if we take this one out of here and do it incorrectly, yes, it's still the same genetic information, but you might get confused when you're going through here looking for the phenotypes. You might get confused because of those letters lined up the way they are. Okay? So let's not make that mistake and write them the way they should be, in A-B order, or letter order. In this case, we have A and B. Alright? So I'm just going to fill these in here for you. You should be able to do this on your own. Notice I'm doing AB order for every single one. So all of my offspring have the same order of letters, different capital lowercase, in most cases, compared to the parents, but all in AB order. 2 A's, 2 B's. So, again, to know if you're doing this right, all of my offspring have 2 A's, 2 B's. Both of my parents have 2 A's, 2 B's. They gave half the genetic information, 1A, 1B, each one gave 1A, 1B, so that my offspring all have the correct amount of genetic information. Let me show you something, again, another trick that you can double check if you did this correctly. Alright? And here is my foil at the top. If I do my gametes correctly, I should be able to spell that out across the top and down the side. If I do them in this order, the way that you see them here, I will have a really easy way to double check to see if I've done it correctly. Okay? I should get a big triangle of genotypes that all share the same phenotype. In other words, each one of these in the orange triangle have at least a big A and at least a big B. Therefore, they share the same phenotype because of the law of dominance. Another double check I can do to see if I did some more of this correctly is I should get this one here, and this one here. Again, makes a triangle, but there's only 3 of them, so here, here, and here are my points of the triangle, but all 3 of these also share the same phenotype. They have a big A and something else, either big A or little a, but they all share the 2 little b's in common. So they have big A and something, 2 little b's. There's 3 of them. I should also have another 3 that are similar, just backwards. Little a's with a big B and a second letter, again, that doesn't matter because of law of dominance. Little a's, little a's, little a's, each having a big B and either a big B or a little b. So again, another triangle check to see if I've done it correctly. My last one doesn't have a triangle check, but it's kind of the runt of the litter, right? Has all the recessive genes, alleles. The only way to have all the recessive, exhibit all the recessive traits is to have all the recessive alleles. In other words, 2 little a's, 2 little b's. Again, so I have a triangle check. 9 that all have the same phenotype. And that is, they are dominant for both traits. They have a big A and a big B. The second letter of each doesn't matter according to law of dominance. I have 3 that have big A's. Law of dominance says big A is all that matters. And little b's. That's these 3 in green. And then I have these 3 in yellow that are little a's, recessive, homozygous recessive, but have at least 1 big B, again, law of dominance. And then again, my last one down here is a 1. So I should get a 9:3:3:1 ratio, phenotype only. We're not too worried about genotype ratio for here because it's a lot more confusing. So I should get a 9:3:3:1 phenotype ratio for this. Okay? But recognize what that means. 9 of them have big A and something that doesn't matter, so I'm just going to put a dash, meaning they could have either or. Okay? Dash meaning they could have big A or little a, doesn't matter. Alright? And the other ones, the B's have at least a big B, could be big B or little b following that, doesn't matter, so I'm just going to put a dash. 9 of them have that. Okay? 3 of them, these in green, have big A and either big or little, following that, but only have 2 little b's. And those are in green. Another 3 have only little a's, and kind of backwards of the last one, a big B with a following number or letter that doesn't really matter because of law of dominance. So here you can see we have variation in opposite corresponding letters, traits, okay? And then finally, the only way to be homozygous recessive for 2 traits is to get 2 homozygous recessive alleles, 2 little a's, 2 little b's. So you can see there's a lot of flexibility here. You can have either big A or little a, big B or little b, and that represents 9 out of 16 in my Punnett Square. You can only get flexibility here for 3 of them, and only, conversely, get flexibility for B. So again, 3 in each of these in the Punnett Square, and then again, kind of my runt of the litter, having all the homozygous recessive traits. So there's my phenotype ratio, and again, you would write this out as 9. Let's say A represents having a cleft chin, and big B represents a straight hairline. Okay, so you would write these out, dominant recessive traits, however they were given to you, but just for an easy example, I didn't use any traits specifically, but that's how you'd write the phenotype ratio. Obviously, you'd have to show the phenotype, whatever that was, if it was given to you. All right? But again, here's the most important part, just really quick. Being able to FOIL out the gametes so that your Punnett Square looks correct, and a quick way to double check, if your parents are 2 A's, 2 B's, each of your boxes, each of your offspring should be 2 A's, 2 B's. And then when I do FOIL correctly, fill in the boxes correctly, I should get triangle check back. First triangle here, 9 out of 16 are dominant. 3 out of 16 are dominant for A, recessive for B. Another 3 out of 16, but now are recessive for A, dominant for B. And then 1 out of 16 is going to be recessive for both. So that's how you do a dihybrid cross correctly. And for phenotype ratio, you will always get a 9-3-3-1.